Take a look at these two plants. Both receive the exact same amount of direct, abundant sunlight, yet the plant on the left is severely stunted, while the one on the right is thriving. If light is the primary fuel for growth, a plant in full sun should, in theory, be flourishing. Finding the cause of this disparity requires looking at the leaf under the microscope. Inside, we find the palisade mesophyll cells, which are packed tightly together to form the plant's primary site for energy production. The leaf is physically optimized for this task. It has a broad lamina to maximize surface area and a transparent cuticle that allows light to pass through while preventing water loss. Deep within the mesophyll cells, the chloroplasts function as biological solar panels, designed specifically to capture and convert incoming light. Since the physical structure of the leaf is already perfected for energy capture, the failure to grow suggests that the limitation lies within the internal process itself, rather than the leaf's anatomy. Photosynthesis relies on raw materials in very specific proportions. 6 molecules of carbon dioxide and 6 molecules of water are required to produce 1 molecule of glucose and 6 molecules of oxygen. Chlorophyll, the pigment inside the chloroplast, captures the light energy needed to drive this entire conversion. These molecules follow distinct physical paths. Water moves up from the stem and into the leaf cells, while carbon dioxide diffuses from the surrounding air through the leaf's protective layers to reach the chloroplast. When light energy strikes the chloroplast, the reaction triggers. The plant stores that energy in a single glucose molecule to fuel its growth and releases the 6 oxygen molecules back into the environment. This chemical reaction depends on the simultaneous availability of all these components. If even one raw material is in short supply, it throttles the entire system, regardless of how much light or water is available. This is best understood through the analogy of a narrow straw. You can have a gallon of water ready to pour, but the diameter of the straw dictates exactly how fast that water can move. In biology, we call the narrowest point the limiting factor. We can see this bottleneck on a graph. The x-axis represents light intensity, and the y-axis represents the rate of photosynthesis. Initially, as light intensity increases, the rate of photosynthesis climbs steadily, producing more glucose for the plant. Eventually, the line levels off into a plateau. At this point, the rate of photosynthesis stops responding to additional light. The plant has reached a limit because it has run out of a different requirement, such as carbon dioxide, which is now the factor restricting growth. This graph compares the same plant species at 20 degrees Celsius and 30 degrees Celsius. Look at the lower line. Even at maximum light intensity, it plateaus much sooner than the upper line. Take 5 seconds to identify the limiting factor for that lower plant. The limiting factor here is temperature. Even with intense light and plenty of carbon dioxide, the plant's internal enzymes cannot work any faster in the cooler environment, setting a hard cap on its productivity. Increasing the rate of photosynthesis requires optimizing the full set of requirements: light, carbon dioxide, and temperature simultaneously to move past that plateau. Experimental proof of these factors requires a specific preparation called destarching. By placing a plant in 48 hours of darkness, we force it to use up its stored energy reserves. This ensures that any starch detected at the end of the experiment is brand new. To isolate the effect of a single variable, we place the destarched plant in a sealed jar with a chemical that absorbs carbon dioxide. This creates an environment where we can test if the gas is truly indispensable for the process. An iodine test on these leaves remains brown, confirming no starch was produced. Without that specific carbon dioxide input, the chemical reactions simply cannot occur. Exam mastery involves connecting these core concepts: how the physical structure enables the process, how the chemical equation maps the flow of matter, and how limiting factors set the maximum rate of growth. Take a screenshot of this summary for your revision. Understanding the why behind these mechanics is the key to handling O-level biology questions. When you're in the exam, remember to use these diagrams and clearly label your inputs and outputs to show how the system balances itself.